and welcome everyone to this podcast for genetics. This will be our last podcast in the group of podcasts that covered mitosis and meiosis. And in this podcast, we're going to focus in on that end product of meiosis. And so we're going to start by talking about gametogenesis. And if we could come up with a quick definition here of gametogenesis, and it's just the meiotic pathway that leads to genetically diverse gametes. So then what are gametes? We can think of gametes as being mature haploid cells used in sexual reproduction. Okay, gametes can be classified in different ways. We can classify them as either isogamous or heterogamous. Species that make gametes that are isogamous have gametes that appear the same. Some examples of this would include fungi. Species that have heterogamous gametes, they have gametes that appear structurally different. Examples of this would be animals and plants. What I'd like to do now is to show you some pictures of gametes that are isogamous or gametes that are heterogamous, and then we'll come back to this board. All right, now that I've defined isogamous and heterogamous, I want to show some examples. Fungi offer a great example of isogamous. We have Aspergillus spore here. Each of these spores are a haploid spore that has been produced through the process of meiosis and then have matured to have this very thick outer coating to protect them. But inside of them is just a single haploid genome, one copy of all their chromosomes. Same thing here with Saccharomyces cerevisiae. They hold all their spores inside of a single structure called a tetrad. But each of these four spores look very similar to each other, but inside of each of these four spores is a single haploid genome. So just one haploid genome in each of these. Even though they look morphologically the same, again, remember, inside the cells, genetically, they're quite different. And fungi don't have males and females. They do have different mating types. So, for instance, they have in yeast here, they have an alpha mating type and an A mating type. So two of these spores will be alpha and two will be A. So that when it does germinate and these spores are released, they can mate with their sister spores here. Now, you're probably more familiar with heterogamous, such as humans' sperm and egg. You can see, unlike the fungal spores here, these are morphologically quite different. The sperm has the head of it at the top here and then a long flagellum. Inside of the head of the sperm is the haploid nucleus. I can put one in here. The oocyte, the egg, is also haploid, but you can see it looks much different and it's larger. In fact, proportionally, it's even much larger than what we see here between these two cells. The sperm has just been enlarged to see it a little bit better. So that's what heterogamous is. They look different. I also want to just take a moment here to talk a little bit about the fact that not all sperm look the same. Not all eggs look the same. But there are some really noticeable differences in sperm. The first example is what we see in the soil nematode called C. elegans. We'll work with that in the lab later on. But these are sperm, and the thing you'll notice is that they lack this flagellum. They are non-motile. Turns out that C. elegans are hermaphroditic, so their sperm are produced right next to their egg, so they don't need to swim. This is a particular species of Drosophila, which has the longest flagellum known. So this is the length of an adult fly, and here's the head of the sperm in here somewhere, and you can see how the flagellum wraps around here, all the way around here, and to the end here. So it's an extremely large flagellum, just to highlight some of the differences between them. Now that we've classified gametes as either being isogamous or heterogamous, let's talk about specific gametes. And I'm going to show just three here. Gametes can be spores. An example of an organism that makes spores as their gametes would be fungi. Fungi are typically isogamous. Now, two types of gametes that we're more familiar with would be sperm and ova. Sperm and ova are both made by animals and plants. Now, all these gametes, as we said up here, are going to be used in sexual reproduction. What I'd like to do now is to begin talking about the formation of sperm and ova in animals. And then we'll come back and talk about sperm formation and ova formation in plants and talk about how they're a little different. So we're going to focus initially on animals. And for the most part, the examples I will talk about will be most similar to humans. Within animals, there's going to be some variations to what I'll describe here, but in general, it's a pretty consistent process. So first, let's talk about spermatogenesis. This is the meiotic pathway in males that produces genetically diverse haploid sperm. I'm going to erase this so I have the whole board to draw out the pathway of spermatogenesis. So first, let's start off with a diploid cell, a unique diploid cell called a spermatogonium. Spermatogonium are one of the two types of cells that can do something pretty unique. They can undergo either mitosis or they can undergo meiosis. No other cells besides the oogonium, which we'll talk about in a moment, can do both of these pathways. It's important that spermatogonium can undergo mitosis so that they can continuously regenerate new spermatogonium. However, when they commit towards making sperm, the cell commits to a meiotic cell division pathway. As they commit to this meiotic pathway, they are called a primary spermatocyte. At this stage, they are still 2N. They will undergo meiosis 1, and then they will produce 2 haploid cells using the same meiotic pathway that we talked about on the previous podcast. These are now called secondary spermatocytes. Secondary spermatocytes will now undergo meiosis 2. And they will produce, each one of these undergoes meiosis 2, so together they will produce 4 haploid cells. At the end of meiosis 2, the products here are called spermatids. What happens next is a process that we'll call maturation. And at this point they mature into, each one of them will mature into a sperm. Because of meiosis, they are genetically diverse. But these 4 sperm that are produced from this one diploid cell are also all similar sized. One thing I'd like to point out as we look at the sperm here is that the genome that is packaged into each of these sperm heads here is packed in here very efficiently. Very little cytoplasm or organelles come in here. In fact, in particular, all of the mitochondrion from the sperm is located right here within the base of the tail of the sperm and the base of the flagellum. This mitochondria provides the energy necessary for movement, but none of the paternal mitochondria or the paternal mitochondrial DNA make it into the ova once it fertilizes the ova. Some other things I want you to know is that sperm are produced continuously from puberty. So once a male hits puberty, they will continuously make sperm throughout their lifetime. In that course of that lifetime, it is estimated that 500 to 600 billion sperm are produced. Per ejaculate, 40 million to 1 billion sperm are released. I point that out because out of all these sperm that are produced, very few are used. At least very few are used during fertilization. Why is it necessary to make so many sperm when you only use few of them during fertilization? And in class, we may have time to talk about that. Okay, now I would like to move on to oogenesis and talk about how ova are produced and talk about how that is quite a bit different than the way sperm is produced. Okay, so let's talk now about oogenesis. So oogenesis is the meiotic pathway in females that produces genetically diverse haploid ova. Alright, now I want to draw out the pathway for oogenesis, highlighting some of the similarities and differences we see when compared to spermatogenesis. Okay, so remember we're going to talk about oogenesis. We need to start off with a cell similar to what we started off with in spermatogenesis, except this time we're starting off with an oogonium. Oogonium are diploid cells, and just like the spermatogonium, the oogonium also has the unique feature of being able to undergo mitosis, or it can commit to meiosis. Just like with the spermatogonium, this is important so that we can produce new oogonium. Now, as we'll see in a moment, all of the oogonium commit to meiosis during fetal development. So it's not like it is with spermatogenesis, where we have to have a constant lifetime supply of them. But during fetal development, it is important that we can keep making new oogonium so that a healthy supply of oocytes can be produced. We'll come back to the timing of oogenesis in a moment. So once this oogonium commits to meiosis, just like in spermatogenesis, the oogonium commits to meiosis and enters prophase 1. And at this point, it is called a primary oocyte. And at this point here, it's in prophase 1. Now each primary oocyte will complete meiosis 1, producing one secondary oocyte and a much smaller polar body. Now, unlike spermatogenesis, oogenesis produces two disproportionately sized structures here. The first is the secondary oocyte, which is much larger, and the polar body, which is much smaller. The secondary oocyte will now complete meiosis 2. Just like with meiosis 1, the products are also two disproportionately sized cells. The first one here, the larger one, is the ovum, and it will also produce a second polar body. Often these polar bodies just disintegrate. I forgot to mention up here, I should go back and do so, that's a haploid copy here, and also in the polar body, these are also haploids. The ovum is also haploid. This ovum now that is produced can now be fertilized, and as it fertilizes this egg, only the head of the sperm enters. The tail falls off. And as I hinted to on the last whiteboard, since the mitochondria are in the flagellum, the tail of the sperm, none of the paternal mitochondria enter into the oocyte. Now the oocyte is full of mitochondria, tons of mitochondria, chock full of mitochondria. And so I want to talk a little bit more about everything inside of this ovum. The ovum contains the organelles. It contains maternal RNA molecules and maternal transcription factors. These are both really important during the early stages of development, that it has these transcription factors and these RNAs ready to begin to make proteins necessary for cell division. Because once this ovum is fertilized, I should mention once it's fertilized, it becomes a zygote. Once it becomes a zygote, it has to divide really fast. And to help that out, it contains these maternal RNAs and transcription factors, plus all of the mitochondria that is also present here, supplying the energy necessary for early embryonic development. Now, I also wanted to touch on one other idea here, and that is the timing of oogenesis. So unlike spermatogenesis, which occurs continuously, oogenesis has a very unique timing. It begins as a fetus and stops, still as a fetus, at meiosis 1. So this would be our stop point. So while she is still a fetus, the primary oocyte is paused, and it will remain paused until puberty. At puberty, each month, one primary oocyte is released and reinitiates meiosis. Now, meiosis won't be completed. Now, I also want to say that meiosis 2 isn't completed until fertilization. Once the sperm enters the cell, the meiosis II will complete. Now, the next thing I want to talk about on this whiteboard is the question of how many are made. So we made a point of talking about that with sperm production, so now let's talk about that with ova production. At birth, there are greater than 1 million primary oocytes. At puberty, there's only about 400,000 left. So in the process of producing the primary oocyte to actually being able to use them for the first time, about 600,000, over half of them are lost. Now over a lifetime, only about 400 are used. So these 1 million that are produced by birth, during a course of a lifetime, only about 400 of them are used. It's an interesting question why it has been selected for a pathway that produces so many potential ova, but only uses 400. Again, that's a question that we don't really know, but we may talk about possible answers to that in class. Okay, now let's move to plants. So we're going to talk about spermatogenesis and oogenesis in plants. Now plants do some pretty interesting things during meiosis to produce their gametes. In fact, they're so interesting, I think they kind of put animal gamete production to shame. They're very unique. But first, let's talk a little bit about the alternate generations of a plant. And let's begin by talking about a zygote, which is formed, as we'll see later on, in a similar manner as a zygote from animals is made, by the union of a sperm and an egg. This zygote will undergo mitotic divisions and differentiations, much in the same way an animal would. Except plants, of course, are going to make a plant. So here's my lame attempt of a flowering plant. We call this the sporophyte stage. And it is dominated by multicellular diploid cells. And in a moment we'll go in great detail about this stage here. As the plant matures, it will make gametes and enter into this meiosis stage. Okay, we will go in greater detail on the next whiteboard, but for right now what I'd like you to know is that this flowering plant contains both male and female reproductive structures. And as such, it will produce sperm and ova. The sperm develop from microspores, and the ova form from megaspores. And this process of going through meiosis and producing sperm and ova we will talk about on the next whiteboard. But for now, the sperm and the ova will fertilize, producing the zygote to carry on the next round of the life cycle. We call this part of the life cycle the gametophyte stage. It is characterized by single cells, single haploid cells. Okay, now what I'd like to do is to expand this part here and talk about how it's going to make both microspores and megaspores and how those ultimately become sperm and ova. Okay, I'm going to start with the same plant, but we're not going to talk about the sporophyte stage. We're only going to talk about the gametophyte stage. I want to remind you that this plant, because it contains both male and female reproductive structures, is able to make both male gametes, sperm, as well as female gametes, ova. Within the male reproductive structures is this structure called the stamen. The stamen contains cells called microsporocytes. These cells are diploid. The microsporocytes will undergo meiosis. Through this process, it will produce 4 haploid, so 1N, microspores. This is where it starts to get a little different compared to animal production of sperm. Each of these microspores, which I'll just draw like this, undergoes mitosis. And within this new structure that's formed, which we will call a pollen grain, we'll find 2 haploid nuclei. So each of these spots is a 1N nucleus. One of these is important in making the pollen tube, which we'll talk about in a moment. The other one will become the sperm that will fertilize the egg. Now, this pollen grain, prior to fertilization, will form a pollen tube. And as it does this, it will undergo mitosis once again. Not the pollen tube nuclei, but the sperm nuclei will undergo mitosis to produce 2 haploid nuclei. So each of these nuclei here are 1 N. And we'll come back to those in a moment after we've made the ova. Remember, this was the pollen tube nucleus, so it's right here containing the information to extend the pollen tube. Okay, now we're just going to wait a moment and leave this pollen tube hanging out here until we finish making the ovum. Now, part of the female reproductive structures is the pistil. Within the pistil is the ovary. Within the ovary are cells called megasporocyte. Like the microsporocyte, these are also diploid, 2N. They will also undergo meiosis, 1 and 2. Just like over here, I didn't mention 1 and 2, but it undergoes both of them. That's how we make the 4 haploids. And as a result, we make 4 haploid megaspores. So they're all 1N. Now this is similar to what we see in animals, in that only one of these will be large enough to survive. The others are small, like the polar bodies we saw in animal oogenesis. So we end up with just this 1 haploid megaspore that can be used in the next steps. Now what this will do, and this is where we start departing from animals, it will undergo 3 rounds of mitosis that will result in a cell that contains 8 haploid nuclei. So 8 haploid nuclei. Now this cell here with the 8 haploid nuclei does something interesting again. These 8 nuclei will now be partitioned into separate cells. And what it makes here is a structure that looks something like this, where 2 of these nuclei form a cell in the middle here. And these we're going to call polar nuclei. Of these other nuclei, one of them ultimately become the embryo. Okay, now that we have our 2 parts here, now fertilization can occur. So what happens next is that the pollen tube will grow into the egg case here. And once it does that, it will insert the nuclei, these 2 sperm nuclei, into this egg case. And the result is a structure that looks something like this, where one of these sperm nuclei will fertilize these polar nuclei here. And I'm going to draw the polar nuclei on the female side in blue, and the nucleus from the sperm nuclei in purple. What we end up here is a 3N nucleus. The other sperm nuclei will fertilize one of these oocyte nuclei. And I'll put the sperm nuclei in purple and the oocyte nuclei in blue. This process is called double fertilization, where one of the sperm nuclei fertilizes the polar nuclei of the egg case here to become this 3N nucleus. The other sperm nuclei fuses with the oocyte nuclei, producing a diploid nucleus. I'm running out of room here, but I want to make sure I close on something here, and that is that the reason we have this double fertilization is that this structure here, this 3N nucleus, goes on to produce the endosperm. The endosperm will provide protection and nutrients for the developing embryo. And with that said, this down here, this 2N nucleus, will go on to make the embryo. By having a double fertilization event here, we've created 2 sources of genetic material. One that forms the endosperm, and one that will form the developing embryo. And as this develops, what will happen is it will form a seed, and the seed can last a very long time until eventually it will germinate to produce the plant. Okay, to quickly summarize, the plant has male and female reproductive structures. They contain microsporocytes and megasporocytes that through meiosis 1 and 2 will produce haploid microspores from the male reproductive structures and haploid megaspores from the female reproduction. They both now undergo mitosis. The male microspores initially undergo one round of mitosis, producing two nuclei. One, which will go on to be the sperm nucleus, that will go on to fertilize the egg, and the other is a pollen tube nucleus that is going to be important in forming the pollen tube. Before it forms the pollen tube, the sperm nucleus will undergo one more round of mitosis, producing two haploid nuclei. Coming back up here to the megaspores, it will undergo 3 rounds of mitosis to produce 8 haploid nuclei. The haploid nuclei are partitioned off into separate cells. It produces a 2 nuclei structure here that we call the polar nuclei, and then 6 haploid nuclei. The pollen tube will transfer 2 of these sperm nuclei into this egg case. One of the sperm nuclei will fuse with the polar nuclei, shown here, that will go on to form the endosperm. The other sperm nucleus will fuse with one of these oocyte nuclei, producing a diploid nucleus that will go on to form the embryo. The embryo will divide and grow using the nutrients and protection from the endosperm as it forms the seed. And in the seed, this embryo will remain until it is germinated to allow the production of the plant. What I've described for you here on the past 2 slides are what we see with angiosperms. Just like with animal gamete development, I didn't talk about all the animals. I mainly focused on mammals and in particular humans. In plants, we focus primarily on angiosperms. There are differences as you move between the different plants, but this is a good taste for what happens in plants. I think we're going to end this podcast here. If you have any questions at all, please make sure you come see me. If not, I'll see you in class. Bye.